Good afternoon, students. I will wait for a minute. Good afternoon, students. Again, I will wait for a minute for the other students to join. Okay, I'm going to start the lecture now. Hello. Good morning, students. Good afternoon, students. Again. Okay, today I'm going to discuss about the anaerobic gram-negative bacteria. Gram-negative bacteria, cocci. Okay, and the phototrophic bacteria also. Okay. Now moving forward with the anaerobic gram-negative cocci, some of the examples are Acidomycoccus fermentans, Megasphaera, Centrococcus, Sacromutans. Okay, and basically it consists of a family Velionolaceriae, and which is grouped under the category which consists of non-motile cocci, which normally occurs in the pairs. Okay, and they are normally found in the oral cavity, respiratory tract, or the intestinal tract of the humans, ruminants, rodents, and pigs. Okay, so I have taken one of the bacteria which is a Veillonella in this group. Okay, and they stain gram negative and a cocci with a size range from 0.3 to 0.5 micrometer in diameter and appear by light microscopy as diplococci, as they occur in chains mostly and also in the short chains. And the specialized structures are basically they are non-sporulating, chemoorganotrophic, in which pyruvate, lactate, malate, fumarate and oxaloacetate are fermented, and carbohydrates and polyols are not fermented except for the one species where fructose fermentation has been detected. And as we are discussing the anaerobic group, okay, Veillonella is also a anaerobic cocci, and the pH, they have a pH optima around 6.5 to 8, and with the optimum temperature requirement of 30 to 37 degrees Celsius, and nutritional requirements are complex and the carbon dioxide is required for the growth, and products basically which they produced are acetate, propionate, carbon dioxide, hydrogen are produced from the lactate. They are oxidase negative, catalase negative, but some species do produce a typical catalase which basically lacks a porphyrin in the structure. And the habitat, they are parasitic in mouths and in the intestinal and respiratory tracts of man and other animals, as I told you earlier. And pathogenicity, they are parasitic to human beings. as well as various other ruminants. Okay, this one is a photograph of a Veillonella, okay, cocci. They are very small in size. Moving forward with a next group, which are phototrophic bacteria, okay, especially phototrophic anoxygenic bacteria, okay, and the phototrophic anoxygenic bacteria are basically divided into four groups, okay, in which one is green sulfur bacteria, next one is green non-sulfur bacteria, next one is purple sulfur bacteria, and purple non-sulfur bacteria. In general, you all know phototrophs are those organisms which carry out photosynthesis, and they capture energy from light and use this energy to manufacture the carbohydrates from the carbon dioxide. Okay, so the photosynthesis occurs in green and purple bacteria under anoxygenic terms, and cyanobacteria in the case of oxygenic terms, as well as you already know algae and higher plants also have a photosynthesis process. And the photosynthetic bacteria which probably are evolved early in the evolution of the living organisms, and uh, perform their own version of photosynthesis in the absence of oxygen. However, algae and green plants make much more of the world's carbohydrate supply. Okay, so there are basically two types: oxygenic phototrophic bacteria and anoxygenic phototrophic bacteria. So let us first discuss some of the basic characters of the phototrophic anoxygenic bacteria. They are mostly gram negative, and cells are spherical, ovoid, and are also sometimes long rods, or some are even spirally shaped. Okay, if motile, they are motile by polar, subpolar, or the peritrichous flagella, or are non-motile, or are non-motile, and none of the described species contains gas vacuoles, and the internal photosynthetic membranes are continuous in the cytoplasmic membrane and consist of a finger-like intrusions such as vesicles or lamellae. Polysaccharides, polybeta hydroxybutyric acid, and polyphosphate have been also found. Okay, whereas in the case of plant cells, normally photosynthetic membranes are basically stacked, whereas in the case of these bacteria the membranes are continuous. Okay, they divide by the binary fission or show polar growth and budding, and color of the cell suspension is green, okay, or maybe yellowish green or yellowish brown or brown red or purple violet. Okay, it is due to the presence of various photosynthetic pigments as well as carotenoids which are present in this photosynthetic bacteria. Under anaerobic conditions in the light, all species grow as photoheterotrophs with various organic substrates also, or as photoautotrophs, either molecular hydrogen, and in some species they also use sulfide, thiosulfate, or elemental sulfur as electron donor, and carbon dioxide as sole carbon source also. So under the oxygenic condition ranging from microaerobic to aerobic conditions in the dark, many representatives can grow chemoheterotrophs also, and some can also grow chemoautotrophs also. In the most species, the formation of the pigment and of the internal membrane systems are repressed under aerobic conditions, but become deep repressed at oxygen tensions below a certain level. Okay, and what do they require for their growth? They require various vitamins, okay, and most common required are biotin, thiamine, niacin, and para-aminobenzoic acid. And these compounds are rarely needed by species such as from the group Chromaceaceae and Ectothiorhodospira, which may require vitamin B12 as their sole growth factor. And growth of the most species is enhanced by small amount of yeast extract, and some species have complex nutrient requirements. Okay. Now moving forward with the unique features, as I told you, okay, they do not have chlorophyll similar to that of plant, but they have a bacteriochlorophyll, okay, which may range from bacteriochlorophyll A to G, okay, from A to G, okay, and these pigments are basically located around the cell membrane, and which are basically continuous in structure, and the habitat, basically members of this group are widely distributed in nature and found not only in the freshwater, marine, hypersaline environments, but also in the moist soils, paddy fields, etc. They live preferably in the aquatic environments with a significant amount of soluble organic matter, low oxygen tension, and rarely found form a colored blooms, and they are characteristically formed by a representative of the other families of the Anoxygenic phototrophic bacteria. Okay, so these are the just the basic features of the phototrophic bacteria. Okay, now moving forward further for the purple bacteria and green bacteria. Okay, the purple bacteria, which contains internal sulfur granules. Okay, what does it indicate? That in their structure they have some sulfur granules which can be easily identified in the microscope. If you want to study this photosynthetic bacteria, you need to use a basically Winogradsky column. You need to use a Winogradsky column for the study. Okay, and it is the best way to study this column. Okay, now moving forward with the examples of purple bacteria. Okay, with the purple bacteria. Okay, in purple also there are two types: purple sulfur bacteria and purple non-sulfur bacteria. Purple sulfur bacteria and purple non-sulfur bacteria also. Okay, and the examples of purple bacteria with the internal sulfur granules are Amoebobacter, Chromium Lamprobacter, Modesto Halophilus, Lamprocystis, Roseopersinia, Thiocapsa, Thiocystis, Thiodictin, Thiopedia, Rostia, Thiospirillum, Genescens. And the external sulfur granules containing purple bacteria is just in one example, Ectothiorhodospira, and the bacteria with no granules, non-sulfur, Rhodobacter, Rhodocyclus, Rhodomicrobium, Vanilli, Rhodophylla, Globiformis, Rhodopseudomonas, and Rhodospirillum. Okay. Now green bacteria with the sulfur granules. Okay, green bacteria with the sulfur granules are Anachloris, Per, Philippi, Chlorobium, Chloroherpeton, Thalessium, Paleodicton, and Prosthechloris. And with no sulfur granules are Heliobacillus mobilis and Heliobacterium chlorum, and with the multicellular filaments are Chloroflexus, aurantiacus, Chloronema, Heliothrix originaensis, and Oscillochlorus Okay, so now what is the basic difference between the photosynthesis in the case of animals, in the plants, or with the other bacteria? Okay, normally in the case of photosynthesis, in the case of plants, oxygen is produced. Okay, whereas in the case of anoxygenic photosynthesis, oxygen is not produced. And there are also other features, and there are also the other features of this bacteria. Okay, they have various water insoluble carotenoid pigments also, which can trap and absorb light energy and transmit it to the bacterial chlorophyll also. and this anoxygenic bacteria grow phototrophically only under the anaerobic conditions and are incapable of forming oxygen. Okay, as I told you. Okay, so one of the bacteria which is basically an anoxygenic, which undergoes an oxygenic photosynthesis, basically which are basically cyanobacteria. Okay, it can produce oxygen, but not this bacteria, purple or green bacteria. Okay, one another difference is with the spectrum. Okay, with the spectrum of photosynthesis, where the photosynthesis occurs in this bacteria. In the case of bacterial chlorophyll, which is basically a pigment found in this bacteria, okay, can absorb light in a long wavelength of about 725 to even 745 nanometer, okay, which is basically in the far red region also. Okay, so the variation basically is present when we compare above with the plant one also. Okay, normally if we were known for the chlorophyll A, which is basically present in the plants, okay, which is basically present in the plants, have an absorption spectrum, okay, which is also known as, or to be specific, absorption maxima with a range of 680 to 685 nanometer, but uh, in the case of bacterial chlorophyll A, it is around 850 to 910 nanometer. Okay, whereas in the case of bacterial chlorophyll B, it has 1020 to 1035 nanometer also. Okay, and with the bacterial chlorophyll C, it ranges from 745 to even 760 nanometer also. And in the case of bacterial chlorophyll G, it ranges from 725 to 745 nanometers. Okay, and the bacterial chlorophyll E has a range from 715 to 725 nanometer. Okay, so you may clearly see a striking difference between the absorption spectrum when we compare with the plants also. Okay, now I am going to discuss a bacteria which is basically a Thiocapsa, which is basically a Thiocapsa. Thiocystin gram negative, cells are spherical. To avoid 1.2 to 3 micrometer in diameter, and tetrads are also formed. Okay, they are non-motile and contain internal photosynthetic systems of vesicular or tubular type, in which the photosynthetic pigments bacterial chlorophyll A or B and carotenoids are located. They do not contain gas vacuoles, and an animal sulfur transiently stored inside the cells. Tropism, as we are studying phototrophic bacteria, okay, they are phototrophic, okay, and maybe chemoautotrophic also, or even mixotrophic. Okay, what do you mean by mixotrophic? Mixotrophic are those organisms which use both the type autotrophy, heterotrophy also, where phototrophy, okay, it can use a variety ones. Okay, under microaerobic to aerobic conditions in the dark, and capable of photolithoautotrophy also with the sulfide or sulfur as electron donors, and the final oxidation product is the sulfate. Now this one is a photograph of a Thiocapsa roseopersonea. A sulfate oxidizing non-oxygenic phototroph, and contains intracellular sulfur granules, bundled tubular pigment vesicles. Okay, now this is a photograph of a colony of Thiocapsa, and this one is a photograph, okay, of a cell of the Thiocapsa, where you can see sulfur granule, okay. So the Thiodicton, now which I am going to discuss, is basically again gram negative, okay, sometimes appear spindle shaped and may form aggregates The cells are arranged end to end in an irregular net-like structures. These bacteria have particularly a net-like structures, okay, and motility, they are non-motile under all conditions and contain similar type of structures which are normally found in the photosynthetic bacteria, okay, contain large irregular shaped gas vacuoles in the central part which are basically not found in all photosynthetic bacteria, and during sulfide oxidation globules of elemental sulfur are transiently stored in the gas vacuole and free peripheral part of the cell. Okay, tropism, they are obligatory phototrophic capable of photolithoautotrophy only with the sulfide or sulfur as the electron donor, and they are strictly aerobic, whereas Thiocapsa can survive in the aerobic condition also as it was a mixotrophic, and normally they are found in mud, stagnant waters of ponds, lakes which contain hydrogen sulfide, okay, but not uncommon in as planktonic bacterium in the sulfide containing hypolimnion of the freshwater lakes. Hypolimnion is a place where there is a less amount of oxygen, okay, and less amount of light, and the final oxidation product will be sulfate, okay. Thiocystis, next bacteria is Thiocystis. They are gram negative, cell spherical to slightly ovoid in shape. They, before cell division, they are diplococcus shaped. Cells occur singly or in pairs or may grow in a irregular aggregate surrounded by a slime. And motility, they are motile by means of a single flagellum and contain photosynthetic membranes of vesicular type, for vesicular type in which photosynthetic pigments bacteriochlorophyll A and carotenoids of group 3 and 4 are located. They do not contain gas vacuoles. During the sulfide oxidation, globules of elemental sulfur are stored inside the cell, and storage of polysaccharide, polybeta hydroxybutyrate as well as polyphosphates. Under anaerobic conditions, cells are capable of photolithoautotrophic growth with sulfide or sulfur as electron donor. They are facultatively chemoautotrophs under the microaerobic to the aerobic conditions, okay. So the products basically which are formed, okay, the final oxidation again will be a sulfate, and habitat is basically which they are involved is an anoxic, means less oxygen containing or without the oxygen containing sulfide water and mud of fresh and brackish water and the marine environments. Okay, this is a photograph of an Thiocystis. Okay, now you can see a diplococci like structure here. Okay, yeah, it may be not clearly visible to you, I think. Okay, so basically purple phototrophic bacteria, okay, these have basically pigments bacteriochlorophyll A and B, okay. Now the next bacteria of the same phototrophic group which is basically an Paleodictin. Gram negative, cells are rod to ovoid, occur singly or in net-like or more or less spherical aggregates. Motility, they are non-motile, and the specialized structures basically branching may occur as a result of a ternary fission. Cells regularly contain gas vacuoles. Cultures or cell materials is either green or brown. Photosynthetic pigments are located in the cytoplasmic membrane, and the chlorosomes which underlie are attached to the cytoplasmic membrane. They are photoautotrophic growth with the sulfide or sulfur as the electron donor, Okay, they are obligatory anaerobic with a temperature range of about 15 to 30 degrees Celsius. And basically Paleodictin require sulfur compounds and also bicarbonates, and a number of organic substrates may be also photoassimilated, and bacterial chlorophyll C, D and E occurs as the major photosynthetic pigment. Okay, whereas in the bacteria which we previously discussed, okay, which basically contain bacterial chlorophyll A as the major pigment, but this bacteria contains bacterial chlorophyll C, D or E as a major photosynthetic pigment. The chlorobactin or isoraniaretin is the major carotenoid component, and during the sulfide oxidation, globules of elemental sulfur are formed outside the cells. The sulfur may be oxidized to the sulfate, okay, and the habitat hydrogen sulfide containing water and mode of fresh water, brackish water and the marine environments. Okay, this is a photograph of an paleodictyon. Okay, this is a photograph. Okay, note this one. No? Okay, next one is a chlorobium. Next bacteria is an chlorobium. Again, gram negative, cells spherical, ovoid, straight or curved rod shaped, 0.3 to 1 micrometer wide and 0.4 to 3 micrometer long, and sometimes much longer also. And cells are often united in chains resembling the streptococci or filaments. Okay, or filaments, and curved rod shaped strains may also form long spirals. They are non-motile and exhibit two clearly distinguishable colors. Okay, one of them is grass green and second one is a brown, also chocolate brown. Significantly, these colors can be recognized under light microscope with the bright field illumination. Okay. Now towards the again to the oxygen requirement of the chlorobium, they are obligatory anaerobic. Okay, with a temperature ranges from 20 to 25 degrees Celsius, and hydrogen sulfide containing mud and water of fresh water, brackish water and even marine environments. They require ammonia as a nitrogen source, and molecular nitrogen is also fixed by some of the strains, and in this case bacterial chlorophyll C, D or E are the major photosynthetic pigments. In addition to the small amount of bacterial chlorophyll A, chlorobactin is the major carotenoid component and the trophic photolithotrophic with the sulfide or sulfur as the electron donor. Okay, this one is a photograph of a chlorobium. As I told you, contains brown pigments. Okay, contains brown pigments. Okay. Now moving forward, again with the photographs of the chlorobium. The photographs of the chlorobium. Now the next one is a Rhodobacter. Okay, next one is a Rhodobacter, which belongs to the family Rhodospiralaceae. They are contain basically, they contain purple non-sulfur bacteria, and cultures appear orange brown to purple red under aerobic conditions. Okay, so these are the general properties of the Rhodobacterium. Okay, of the family Rhodospiralaceae also. Okay, and the purple non-sulfur bacteria exhibit a diversity of shapes ranging from helical, okay, non-prosopagate rod shape, ovoid or even spherical shape that multiply by the binary fission or sometimes by budding also. Okay, So they are gram negative, avoid this Rhodobacter. Okay, they have polar flagella. Internal photocytic membranes are present as vesicles. Okay, they are photoautotrophic and is possible in the presence of the sulfide as electron donor, and in some species with the thiosulfate and molecular hydrogen as well. Growth occurs photoheterotrophically under anaerobic conditions in the light with a great variety of organic compounds as carbon and electron sources. The photosynthetic pigments are basically bacteriochlorophyll A and carotenoids of the spiridion series. Okay. Now next one is in Thiospirillum genescens. Next bacterium is in Thiospirillum genescens. Okay. They are gram negative, cells curved or rod-shaped spiral, or may also form irregular aggregates surrounded by a slime. Motility by means of multitrichous polar flagella. Under certain conditions, cells may be non-motile. And specialized structures of Thiospirillum genescens contain internal photosynthetic membrane systems of the vesicular type in which photosynthetic pigments are located, and they do not contain gas vacuoles Elemental sulfur is basically stored in the cells, and the storage materials are polysaccharides, polybeta hydroxybutyrate and polyphosphates. Tropism, they are obligatory phototrophic and capable of chemolithotrophic growth with the sulfide or sulfur as their electron donors, and they are strictly anaerobic in which sulfate is the ultimate oxidation product. Okay. Now these are the various photographs of other species also, which contains Chromatium venosum. Okay, this one Chromatium venosum, which belongs to the family of Chromaceaceae, and the family contains a purple sulfur bacteria, and the cultures appear orange brown to purple violet also from the family Chromaceaceae. And all genera are capable of photolithotrophic growth using, I think, hydrogen sulfide or elemental sulfur as the electron donor for the carbon dioxide fixation. Okay, so when H2 is the electron donor, globules of elemental sulfurs are also found, usually inside the cells. Now check these globules. Okay, now see the black dots which appear here. Are basically globules of the sulfur. Okay, are globules of the sulfur, and the sulfur is eventually oxidized to the sulfate, and some strains can also grow organotrophically also, and most species are anaerobic and cannot grow in the dark even under microaerophilic conditions. Okay, so the next one, which we have already studied, Thiospirillum genescens. This is the structure of the Thiospirillum genescens. Okay, the last one is a Thiopedia rosea, where you can clearly see where the cells are basically stacked with each other. Okay, where the cells are stacked with each other. Okay, now as I told you in the earlier part of the lecture, that this phototrophic bacteria have a variety of pigments, okay, due to which they produce different types of pigments by the pigments. Okay, so this one is a photograph, okay, of three different blooms of phototrophic bacterium. The first one is Thiocystis genescens, the second one is in Chromatium ocani, the third one is the Chlorobium limicola. The third one is the Chlorobium limicola. Okay. Now colored photographs of the group Chromatium ocani, Chlorobium limicola, Thiocystis genescens, Paleodictin, Clotharity for me. Okay, please have a look. I will give you all a half a minute. And yes, I forgot to tell you about the Chlorobium species, which belongs to the family Chlorobiaceae also. Chlorobiaceae basically, and family basically contains green sulfur bacteria, and normally the cells of this family are ovoid, bean shaped or rod shaped. Okay, and multiply mostly by the binary fusion. Okay, and gas vacuoles occur in some of the species of the family Chlorobiaceae, and the green sulfur bacteria live as photolithotropes using hydrogen sulfide as the electron donor for the carbon dioxide fixation. The granules of the elemental sulfur are basically deposited outside the cell, never within the cells. Okay, never within the cells. Okay, and finally they will be oxidized to the sulfates. Okay, they will be finally oxidized to the sulfates. Okay, next one bacteria which is an Chloroflexus. Okay, last bacteria I think of our today's lecture, Chloroflexus. They is 10 gram negative and it belongs to the family Chloroflexiaceae. Okay, the family contains green non-sulfur bacteria and the main genus is Chloroflexus and is thermophilic with an optimum temperature of around 52 to 60 degrees Celsius around. Uh, they are found in the hot springs where it forms green or orange mats. Okay, and they have very long filaments also. Okay, they have a very long filaments. Okay, so Chloroflexus cells are basically have filamentous structures which exhibit basically gliding motility. Okay, they are motile by means of gliding, not by means of flagella, as flagella are not present, and these are basically photoorganotropes. Okay, they are sometimes also photoautotropes also. Okay, some are chemoheterotrophs also, and several carbon sources are utilized, example acetate, glycerol, glucose, pyruvate as well as glutamate. Okay, similar to other bacteria, they can go chemolithotrophically with the hydrogen sulfide as electron donor, but in the dark conditions they can grow aerobically as the chemoheterotrophs also. Okay, now you can clearly see the structure of Chloroflexus, which has a longer filaments, Okay, next one, Ectothiorhodospira. Okay, Ectothiorhodospira, gram negative morphology, cells are spiral or vibrioid or rod shaped. Okay, it is R here, rod shaped, and motile by means of polar flagella with or without gas vacuoles. It may depend from species to the species. Okay, and tropism able to perform anoxygenic photosynthesis with bacterial chlorophylls, carotenoids as the photosynthetic pigments. Okay, oxygen they grow anaerobically. Okay, sulfide is oxidized to elemental sulfur, which may be further again similar to other organisms will be converted into sulfates. And members of the Ectothiorhodospiraceae are found in the marine to extreme saline environments containing sulfide and have a neutral to alkaline pH. Okay, this one is a photograph of an Ectothiorhodospira. Giving okay, I'm going to end the lecture now. Okay, tomorrow we will discuss about the phototrophic oxygenic bacteria. Okay, we will have a lecture on photosynthetic oxygenic bacteria tomorrow, and please join our webex session with the link which I have shared with you. Okay, please join with the webex session with the webex link which I have shared with you. Okay.